I'm Dr. Reena and I'm working as a lecturer in botany department in Government PG College for Women, Sector 14, Panchkula. Today I'm going to discuss with you the genus Phytophthora, and first of all we'll discuss about the systematic position. Phytophthora belongs to division Eumycota, class Omycetes, order Pyronosporales, family Pythiaceae, and genus Phytophthora. First of all, we'll discuss about its occurrence and habit. This genus includes 75 species, and out of which the 15 are reported in India. Most of the species are facultative saprophytes, and few species are facultative parasites. And this genus is considered to be very destructive, as phyta means plant and ophthora means destroyer. It causes many diseases in flowering plants, and the most common disease caused by Phytophthora infestans is late blight of potato. In 1843 to 46, there was a potato famine in Europe, which led to death of 250,000 people due to epidemic caused by Phytophthora infestans. There are other species also which cause many diseases in the plants, like Phytophthora citrophthora. It causes brown rot of lemon. Phytophthora cryptogyia cause root rot of tomato. Phytophthora megasperma causes root rot of cauliflower. Now we'll discuss about the symptoms of late blight of potato. This disease is caused by Phytophthora infestans, and on potato, the botanical name of potato is Solanum tuberosum. This disease is not very common in plains because temperature is higher and there's drought in the potato growing season, but it is common in the hills above 6,000 feet. Now, the symptoms of this disease: hydrotic spots, they appear at the margins and the tip of the lower leaves, and these spots become necrotic and turn brown to black in color. And in moist weather, these spots spread and cover the whole top of the plant. The host plant decays and produces foul smell, and the diseased tubers are depressed and wrinkled. And the upper skin of the tubers become brown and then turn black. Later on, rusty black spots they develop below their skin, and the tubers decay before the harvest and they turn pulpy. So you can see here in this picture the infected potato leaf and potato tuber. So these infected areas are shown on the leaves and the potato tuber. So you can see here these spots are present on the leaves which turn black in color. Now we'll study about the vegetative structure. So it consists of long filamentous, branched, aseptate and multinucleate hyphae. Such condition is known as acellular cenocytic. Mycelium consists of branched hyphae, and septa are not present, but the septa may appear later on during the differentiation of the sex organs. And the mycelium consists of hyphae which grow in the intercellular spaces, but they produce haustoria which enter into the cells to absorb the nourishment from the host cells. And internally, the hyphae, they are not septate, but they contain cytoplasm, and number of nuclei are also present, and the food is stored in the form of oil drops. Now, as far as the reproduction is concerned, so you can see here in the diagram that the hyphae grow in the intercellular spaces, and they produce haustoria which enter into the cells to get the nourishment. Now, as far as the reproduction is concerned, the vegetative reproduction takes place by the formation of terminal or intercalary chlamydospores, and these spores are thick-walled and store reserve food material. They germinate by giving out 3 to 11 germ tubes which develop sporangia at their tips. Now, in asexual reproduction, which takes place by the sporangia formation. Now, how the sporangia are formed? We'll discuss about the formation of sporangia and Phytophthora infestans. So you can see here in the diagram that sporangia are developed during the damp weather. Some special hyphal branches, they emerge out singly or in clusters through the stomata, and they pierce through the epidermal cells. And these branches, they develop, these are known as sprangiophores, and terminally they become globular, or you can say they become swollen to form multinucleate sporangium. Now this sporangium becomes separate from the rest of the branch by septum formation, and after the formation of sporangium, the apex of the sprangiophore continues to grow further, and this pushes the sporangium to the lateral position and produce another sporangium. In this way, the sprangiosspore produce number of sporangia, and this type of branching system is known as sympodial. In this way, large number of sporangia are developed on the sprangiophore. Later on, these sporangia break off from the sprangiophore by wind or rain, and the places of attachment leaves knee-shaped bending in the sprangiophore. Now the sporangia are carried by wind or rain. Now the mature sporangium, this is ovoid or lemon shaped, and there's a small stalk at the base and thin papilla at the terminal end, and it encloses 5 to 8 or more nuclei in the dense cytoplasm. Now we'll discuss about the germination of the sporangium. So sporangium germinate within few hours of dissemination, otherwise it loses its viability. On reaching a suitable host, it absorbs moisture and germinate. Under the moist conditions and the low temperature, around 12 to 15 degrees Celsius, they germinate indirectly by producing zoospores, and they function as zoosporangia. And under high temperature conditions, they germinate directly by giving out germ tubes and function as conidia. So the sporangia are called conidiosporangia. Now during the favorable period, each sporangium absorbs water and swell. Now what happens, that the protoplasm that is cleaved into many pieces, and these protoplasmic uninucleate pieces metamorphose into zoospores. Finally, the papilla will burst open and the zoospores are liberated. The apical papilla may form vesicle, and in this way, the zoospores are released when the papilla will burst open. And the zoospores are kidney-shaped, uninucleate, and biflagellate. They have two flagella. One of them is tinsel-shaped, and other is whiplash type. Now, liberated zoospores they swim in the film of water on the host surface, and first they swim fast, and then they become slow and then stop. Later on, they develop their own wall, and then they germinate and by producing a germ tube, and they develop aprisorium at its terminal end, which help in penetration. So aprisorium is a flattened pressing organ which help the germ tube to penetrate the host through the stomata or the epidermal cells. Now we'll discuss about the sexual reproduction. In the sexual reproduction, it is oogamous type, and the mode of sexual fusion is gametangel contact type. The male sex organs are antheridia, and the female sex organs are archegonia. Now, the species of Phytophthora may be homothallic or heterothallic. So, homothallic means antheridia and archegonia, oogonia are produced on the same thallus, and mycelium. And the heterothallic means the antheridia and oogonia are produced on the different mycelia. Now, depending on the position of antheridium in relation to oogonium, the various species are grouped into two categories: perigynous antheridium and amphigynous antheridium. Perigynous antheridium, the position of antheridium is lateral to oogonium. You can see here in the diagram that the position of antheridium is lateral to oogonium, and amphigynous antheridium, the position of antheridium is different. It lies at the base of oogonium, forming a funnel-shaped collar. For example, Phytophthora infestans. Now we'll study about the development of sex organs in amphigynous type. So you see here in the diagram that the antheridium develops first in the form of terminal swelling of the male branch. You can see here the male and the female branches are present, and the tip of the male branch, it becomes globular and form the antheridium, whereas the female branch pierces through the maturing antheridium. It emerges out on the other side, and antheridium becomes irregular shaped, and to sustain the pressure it develops papillae. It becomes irregular shape, and it can sustain the pressure. So you can see here the developing oogonium, it becomes globular in shape. And you can see here in the oogonium, the cytoplasm is differentiated into two: nucleated dense central ooplasm and vacuolated peripheral periplasm. Now what happens that the nuclei divide in the ooplasm, and finally all except one migrate to the periplasm, where all of them degenerate and only one is left in the ooplasm. Now what happens that the nucleus which is present in the ooplasm divide into two, and the one of them will degenerate and other form the functional egg nucleus. Similarly, the nuclei of the antheridium also degenerate, leaving single functional male nucleus. Now you can see here the mature oogonium, and it is globular structure, and it has central dense ooplasm and peripheral vacuolated periplasm, and in the central dense ooplasm there is a single large egg nucleus is present. And antheridium is irregular shaped, and this is present below the oogonium as a collar shaped structure, and it develops 2 to 3 papillae and single main nucleus is present. Now at the time of fertilization, one of the antheridial papilla elongate and grows towards the oogonium. Now the tip of the papilla that get attached to the side of the oogonium, the common wall between the two dissolves and the protoplasm of the antheridium along with the nucleus migrate into the oogonium. Now after this, first the plasmogamy will take place, the cytoplasm will fuse, and the second stage is karyogamy, in which the two nuclei fuse, and that is one of the male nucleus and the female nucleus, or you can say the egg nucleus and the male nucleus, they fuse and leads to formation of diploid nucleus. Now after the fusion of the nuclei, oospore is developed. Now it undergoes a period of rest. Now the oospore develops its own cell wall and lies freely in the oogonial wall. It is called aplerotic. In some species, the oospore completely fill the cavity of the oogonium. You can see here. And after passing the period of rest, the oospore start germinating. You can see here the germination of oospore. It absorbs water and become activated. The diploid nucleus divide by meiosis just before germination. Now the further nuclear divisions are mitotic, and in this way large number of nuclei are formed. Now the outer wall of the oospore cracks and the inner wall comes out in the form of a tube, and at the terminal end of the germ tube it develops a zoosprangium. So in the zoosprangium, zoospores are developed, and which further behave in the same manner. As you can see here, the zoospores, they are kidney shaped and they have two flagella, and they swim for some time. Later on they develop their own wall, and later on they germinate to produce a new mycelium. So in this way the sexual reproduction takes place, the new mycelium is developed. So this is all about the Phytophthora. Thank you.